Hello my dear students, I welcome you to your favorite channel NEET World. This is Vasavi Ma'am. I welcome each and every student to the NEET Foundation series of class 9th biology. Yes. So students, in this today's particular session we are going to discuss about the top most important diagram based questions from the chapter Cell, the fundamental unit of life. Yes. So students, we have discussed in detail about this first chapter and we have done with it the cell, the fundamental unit of life. Just you can tap on the playlist in our YouTube channel NEET World to get all the concepts clear. Yes, before starting the session, let's have a positive quote here. Education is the most powerful weapon which you can use to change the world. Education is the main tool that can build and that can empower the individual by providing them with the knowledge, skills and values to make a better and bright future, students. Yes. So let's get started with this session. I request each and every student to be please stay tuned until the session gets end, students, because we are going to solve some important NEET foundation questions. Okay. Yes. So in this session we are going to discuss some important cell organelles along with its important labeling parts and functions. So before that we are going to discuss in general what does actually the eukaryotic cell contains. Okay, the eukaryotic cell contains the important membrane bound, non-membrane bound and single membrane and double membrane bound cell organelles. So just have a look here, students. Yes, we have discussed about each and every cell organelle in detail. Just you can go through the playlist in our YouTube channel NEET World. Yes, so a typical eukaryotic cell contains nucleus, yes, next mitochondria and the next one endoplasmic reticulum and next vacuoles in plants and the next one is centrioles in animals, and along with these we are also going to have the Golgi body and many other things. Okay, so let's have a look about all these things in detail. Yes. So students, here just I am going to explain the important labeling parts and its functions along with these particular cell organelles. So here the given cell organelle is a nucleus. Okay, so the nucleus is a double membrane bound cell organelle. The outer and inner membrane it is going to contain. So together it is called as nuclear envelope, which is going to contain outer and inner membrane, and here we have a stained spherical body that is called as nucleolus, which is just involved in the process of making of the ribosomes. And next here the colored network like structures, the colorful network like thread like structures are called as chromatin. These chromatin is going to contain a units called as genes and DNA, which is going to contain the genetic material in them. So these are going to be important to develop the process in each and every individual, students. Okay, so the DNA is going to contain the genetic material which is involved in the process of complete development and growth of the organism. And next here is the fluid filled material that is called as the nucleoplasm, and the complete nucleus is continuous with the endoplasmic reticulum. Can you see this? Yes, this is the endoplasmic reticulum. Yes, so which is where it is going to be attached with the ribosome. So it is the rough endoplasmic reticulum, and here are the nuclear pores. The outer surface of the nucleus is very rough in structure due to the presence of the endoplasmic reticulum and ribosomes on it. And next here we have the nuclear pores. These nuclear pores are involved in transporting some materials like proteins from in and outside to the cells. So these are just the main labeling parts you need to remember along with its function. This is the nuclear envelope because it is going to have the double membrane bonds. Okay, it is going to have two membranes, hence it is the double membrane bond cell organelle, and this is a nucleus which is darkly stained spherical body, and next it is the chromatin. It is a very most important to be remembered, students, because the chromatin contains the DNA which is actually important for the individual to grow and to access some important characteristics, students. And next here it is the nucleoplasm. Yes, and this one is the endoplasmic reticulum where the ribosomes are attached to it, hence it is called as rough endoplasmic reticulum, which is involved in synthesis of proteins. And here these are the nuclear pores which are actually involved in transporting the proteins from in and out of the cell. So this is just about the nucleus. So you have to remember all these concepts. Okay. So at the end we are going to solve the PYQs. So, the next double membrane bound cell organelle is the chloroplast. Yes, it is a double membrane bound cell organelle. That chloroplast is going to have, yes. So, chloroplast is the double membrane bound cell organelle. In the chloroplast, we can find some pancake like structures which are arranged one above the other. So, this is called as grana. And the single unit is called as the thylakoid. Okay. So, grana and thylakoid, and the next here we can find the fluid filled space called as the stroma, and same this chloroplast is going to have its own genetic and circular material, okay, that is called as DNA. This is the outer and inner membrane. So, we have here stroma and grana, and the next one is thylakoids. Okay. When these single pancake like structures, okay, when the single structure is present, it is called as thylakoid. When they are arranged like a stacks upon one on the other, then it is called as the grana. So, here the main thing you all need to remember is the stroma. Okay, stroma is a fluid filled material here. So, this is the main site for the dark reaction. And grana is the main site for the light reaction. And it is also going to have its own circular genetic material, that is DNA. That's it. Okay. So, and in detail about this chloroplast, about nucleus, in detail we have discussed in our YouTube channel. Okay. Just you can go through the playlist Cell the Fundamental Unit of Life. There you can understand each and every cell organelle along with its functions clearly. So, in this today's session, just we are going to solve the diagram based questions. At the end, we are going to discuss how the questions are going to be asked from these particular cell organelles, students. So, just remember the important functions and its important labeling parts. Okay. Yes. And the next double membrane bound cell organelle is the mitochondria. Okay. Mitochondria is called as powerhouse of the cell. Okay. So, I will just give you one interesting facts about the cell organelles. Mitochondria is called as powerhouse of the cell, chloroplast is called as kitchen of the cell, and nucleus is called as brain of the cell, lysosomes are called as suicidal bags of the cell. Okay. So, these all are some just interesting facts to be remembered. Yes. So, this is a double membrane bound cell organelle, that is mitochondria. It is just involved in the generating the energy in the form of ATP. So, the inner membrane, okay, this is the outer membrane, and the inner membrane of the mitochondria is forming into highly folded like structures called as cristae. Can you see this? This is the cristae which increases the surface area of the mitochondria. Yes. This is the inner membrane and this is the outer membrane. So, it is just the sausage shaped. Okay. It gives the appearance like a sausage shaped thing. Okay. Yes, and the inner membrane, the cristae, okay, the cristae is going to have the elementary particles. Okay. It is going to have the elementary particles like F0 and F1. Okay. These are the elementary particles which are just involved in the generating of the energy in the form of ATP, and this is the complete matrix of the mitochondria, the complete fluid. And same as the chloroplast which is going to have its own genetic material in the form of circular DNA, same the mitochondria is also going to have the circular DNA. Clear with the concept? Just small concept here, you need to remember these F0 and F1 elementary particles. Yes. F0, F1 complexes, this. And then next you need to remember its own circular DNA, and the next the inner membrane is called as the cristae. Okay. Cristae is going to increase the surface area of these mitochondria, and the inner membrane is going to contain many enzymes which are actually important for some functions like Krebs cycle, students. Yes. And next one is the prokaryotic cell. A typical prokaryotic cell structure. This is also the very most important important thing I can say to be learned, students. Okay, because this concept is going to be appeared in the class 11th and 12th also. So, I request each and every student to learn the basics in the 9th standard itself. So, it is a typical prokaryotic cell. In the prokaryotic cell, you need to remember mainly the region, the genetic region called as nucleoid. Yes, called as the nucleoid, because here we are calling it as a primitive type of nucleus, because it is actually the undefined nucleus. Undefined nucleus. Why we are calling it as undefined nucleus? Means it is not going to be surrounded by the any nuclear envelope or any nuclear membrane, and it is not going to have any genetic material in the form of like chromatin material, nuclear envelope. These all things are going to be absent. Okay. So, justly the genetic material is just going to be flown freely in the cytoplasm. Hence, we are calling it as an undefined nucleus. Hence, it is called as a nucleoid, because it is not a true nucleus, because it is going to lack the nuclear envelope, chromatin material, nucleoplasm, all these things it is going to be lacked. So, we are calling it as undefined nuclear region. This you need to remember, and one more thing you need to remember is capsule. Okay, capsule, and the next here is the cell wall, and the next one is the plasma membrane. It is a prokaryotic cell which is going to be covered with the cell membrane or plasma membrane, and that plasma membrane is followed by cell wall, and later the extra additional protection, the non-cellulosic layer, that is called as the capsule. This capsule gives the protection to these bacteria against the desiccation and many other things. Okay, we have also discussed its functions in detail. Yes. So, these all are the things you need to remember, and one more interesting thing is the extracellular projections. Okay, these are called as pili. Okay, so these are actually involved in the bacterial reproduction methods like conjugation. Okay, these are involved in the conjugation process, where it is going to transfer its genetic material from one bacteria to the another bacteria. Clear with all the important diagrams and its important labeling parts along with its functions. So, now we are going to solve some top most important 8 to 7 previous year questions of NEET Foundation students. Yes. So, here is a given question. Refer to the given diagram of the animal cell and select the correct options regarding P and Q. Yes. Regarding P and Q, we need to select the correct options regarding to this animal cell. So, before understanding this, we need to know what are the cell organelles, actually the P and Q. Yes. So, here P is the Golgi apparatus, and the Q is rough endoplasmic reticulum. Q is the rough endoplasmic reticulum which is continuous with the nucleus. So, the P is a Golgi and Q is rough endoplasmic reticulum which is attached to the nucleus. So, now we need to select the correct option. So, here are the given options. Option 1, some of the vesicles of P store the digestive enzymes obtained from the Q. Okay. Some of the vesicles formed of the P. Okay. So, what is P? Actually, it is the Golgi apparatus. They store the digestive enzymes obtained from the Q. So, here what it is Q? Q is the rough endoplasmic reticulum which is going to generate the proteins. So, later here you need to understand the concept. Okay. The proteins synthesized from the rough endoplasmic reticulum are going to be packed. Okay. They are going to be packed inside the vesicles, and after packaging, the vesicle with each protein is going to be modified inside this Golgi apparatus, and then later it is going to secrete, and then later it is going to fuse with the lysosomes. So, now here the vesicles of the P store the digestive enzymes obtained from the Q. Yes. Q in inactive state to form the primary lysosomes. Okay. So, here the question is actually here when the vesicle is going to contain the proteins, they are going to be in an inactive state, but when they are going to fuse with the lysosomes, that proteins are going to become the active state. Okay. So, the proteins synthesized from the rough endoplasmic reticulum are going to be synthesized, they are going to be modified and secreted from these Golgi apparatus. So, after secretion of the proteins along with the vesicles, they are going to remain in an inactive state, but when they are going to fuse with the lysosomes, they are going to form the active proteins. Yes. So, this option is right. And next one, option 2, polypeptides synthesized at P are transported to Q. Okay. So, where actually the proteins are going to be synthesized at the Q, but here in the option he is saying that polypeptides are synthesized at the P are transported to the Q. So, this is the wrong option, students. Okay. So, here actually the proteins are going to form the polypeptides, right. So, here Q is going to synthesize the proteins which is going to make the polypeptides, but not the Q here. Yes, but not the P are modified into functional proteins for the secretion. This is wrong statement, because we can frame the option in this way: polypeptides synthesized at Q are transported to the P. Okay. Protein synthesized at the Q, it is nothing but at the rough endoplasmic reticulum are they are going to modify into polypeptides, and then later they are going to be transferred to the Golgi bodies. But in reverse it cannot happen, okay, because proteins are always going to be synthesized from the rough endoplasmic reticulum. Later they are going to be modified and packed inside the Golgi body. So, this option is also the wrong option. Option 3 is Q synthesizes acrosome, which plays an important role in the fertilization process of humans. It is an absolutely and irrelevant option regarding to this particular chapter, cell the fundamental unit of life. So, this is the wrong option, because this we are going to study in the chapter reproduction. Okay. So, this is the wrong and irrelevant option. Coming towards the option 4, P is absent in pancreatic cells and the cells of salivary glands, whereas Q is absent in the liver cells. Okay. What is P here? P is the Golgi body. So, P is absent in pancreatic cells. So, this option is also wrong, because these Golgi bodies and option Q, what it is, rough endoplasmic reticulum, this we can find in the liver cells as well as in the pancreatic cells. Okay. This option is also wrong. So, here he is saying that P is absent in pancreatic cells. No, because P is Golgi body, it is also present in the liver cells, which is involved in the secretion and synthesizing of insulin and many other hormones, etc. So, here among these 4 options, option 1 is the right option. Okay. Some of the vesicles of P store the digestive enzymes. These option P, okay, the Golgi bodies are going to store the digestive enzymes, and when they are going to fuse with the lysosomes, then they are going to become active. So, clear with this first question. This question is completely related with the endoplasmic reticulum, proteins, and the next modification and secretion of the protein, students. Yes. Next, coming towards the second question, refer to the given structure of prokaryotic cell and select the correct statement regarding it. Yes. So, as we have discussed some important labeling parts, now you need to answer what are these given labeling parts here. Yes. So, students, just look here. What is P? P is a capsule. First, let us label all these parts, and next one, what is Q? Yes. Q is a cell wall and R is a cell membrane or plasma membrane. And what is S here? Yes, these outgrowths, these are the pili. So, now by looking into it, let us solve these options, students. See, P, here the first option is P is a layer of non-cellulosic polysaccharides. What is P? P is a capsule which is going to give the protection to the bacteria. Okay. So, it is a non-cellulosic layer, it is a right, and it is made up of the polysaccharides. Okay. The capsule is made up of, we can find it in 2 forms, slimy layer and next one is capsule layer. Okay. So, both of them are made up of the polysaccharides, and this is just an extra protection which is present around this cell wall in the prokaryotic cell, okay, which is present inside the bacterial cell. Yes. And next, coming towards the second option, Q is double layered in gram positive bacteria. What is Q? It is a cell wall. Okay. So, now look into this. Yes. So, regarding these you need to solve the right option, students. So, here Q is a cell wall. Q is the cell wall of this prokaryotic cell. It is gram positive. Okay. It is double layered in gram positive. It is wrong, students, because it is very thin and it is made up of only single layer called as peptidoglycan in the gram positive, but it is not made up of double layered. But whereas it is single layered in the gram negative, it is wrong, students, because in the gram negative it is made up of double layered along with the peptidoglycan and some lipopolysaccharides. Okay. So, in gram negative it is double layered and in the gram positive it is single layered. This is a differentiation in between them. So here the options are twisted, okay, here the terms are twisted, so this is the wrong option. And next, coming towards the option 3, R is an impermeable covering that restricts entry and exit of molecules into and out of the cell. What is R here? Cell membrane, okay, cell membrane is also called as plasma membrane. It is semi-permeable, but it is not impermeable, okay, it is semi-permeable, it is selectively and semi-permeable only to the transport of some molecules and ions, okay, but it is not impermeable in nature. So this is the wrong option. And next, coming towards the option 4, S could be tubular outgrowths that are involved in locomotion only. What is S given here? Yes, can you see here? See here, this yes, outgrowths. Okay, what are these? Just now we have discussed now pili, okay. These are the outgrowths. These are not only just meant for the locomotion, but these are meant for the attachment, and they are mainly involved in the conjugation process, okay. This is the most important process where the bacteria can transfer its genetic material from one bacteria to the another bacteria, okay. These are the main structures where the genetic information is going to be transferred in between the two bacterial cells, yes. So this is also the wrong option, okay. It is not only involved, okay, not only locomotion, it is involved in attachment and in the conjugation process. So among all these, the right option is first option. P is a layer of non-cellulosic layer, okay. What is P? Capsule is a non-cellulosic layer. It is made up of only the polysaccharides, yes. Yes, look into the third question. Identify the cell organelles L and M and select the correct option related to their functions in a cell. So here is a given diagram, okay. So see students, look into this. So in this way you are going to get the diagram based questions, okay. You need to understand the labeling parts as well as its functions mainly, okay. What is L here? So actually it is not visible, I am just going to draw it, yes. So L is a Golgi body, okay. So L is a Golgi body and M is a vacuole, okay. M is a vacuole which is single membrane bond which is going to be surrounded by the tonoplast membrane, and here L is these are the Golgi bodies, okay. So now let us look into this. Select the correct option related to their functions, okay. We need to understand its functions, okay. Golgi body functions and vacuole functions. First option, option L, okay. The labeling part L, formation of lysosomes, okay. And next M is maintenance of maintenance of the cell turgidity. This is absolutely the right option, students, okay. So because in the Golgi bodies, after modification, synthesizing and secretion of the proteins, they are going to be packed inside the vesicles, and then next it is going to fuse to form the vesicles are going to fuse with the lysosomes to form the active lysosomes to digest the other cellular waste materials, okay. So here actually the Golgi bodies are involved, okay. They are involved in process of making of the lysosomes, okay. They are involved in formation of lysosomes, and next here it is a vacuole. Vacuole is going to contain the cell sap, okay. Sap is nothing but which is going to be composed of some fluid substances along with some waste materials, minerals and some other nutrients, okay. So this vacuole is going to contain high amount of the sap, okay, high amount of the water content. So it is going to maintain the turgidity in the plant cells, okay. This first one is the right option. Coming towards the second and third and fourth, 2, okay, option 2, yes, labeling part L, synthesis of lipids. This is absolutely wrong because only smooth endoplasmic reticulum is involved in the synthesis of lipids but not Golgi bodies. So this is the wrong statement. Spindle formation during cell division, this is also the wrong option. This we are going to study in the cell cycle and cell division chapter of class 11th, okay. Now it is not going to be included, students. So it is the wrong option, yes. And next one, option 3, digestion of foreign materials, storing byproducts, okay. Which is involved in digestion of the foreign materials? Lysosomes are involved but not Golgi, okay. So L is Golgi body, it is not involved in digestion of the cellular waste materials, it is just involved in modification and secretion of the proteins. So this one is the wrong. And next, storing byproducts, okay. M, vacuole, okay. It is just going to store some of the waste products, okay. But this M is right. and L is wrong. Okay, but we have to match the right pair. So, this complete option is going to frame a wrong statement. So, coming towards the option 4, synthesis of proteins, production of ATP. Which is involved in the synthesis of protein, students? Rough endoplasmic reticulum, right? But here production of ATP, in the sense here you need to label the part called as mitochondria, but here the given options, the labeling parts are only two. One is Golgi bodies and second one is vacuole. Okay, here the labeling parts, in the labeling parts we cannot find the mitochondria. So, among all these we can find only the right option, first one. Okay, formation of lysosomes and maintenance of cell turgor. Okay, so this will be the vacuole and it is the Golgi body. So, the right option is option 1. Yes. So, students, please be patient and try to listen complete session. These are the most important questions, most important diagram based questions. So, these all the concepts you are going to learn in your 11th and 12th standard, students. So, please try to build a strong foundation in the 9th standard itself. Yes, refer to the given figure of the cell and select the correct statement regarding it. Okay. So, this is a given eukaryotic cell. So, just here I think the labeling parts are not visible to you. I will just make a rough diagrammatic view of it. Yes. Yes. Just a minute, students. Yes. So, this is the rough endoplasmic reticulum which is continuous with the nucleus. This is the nucleus. Okay. So, this is the rough endoplasmic reticulum attached with the ribosomes, and here the X, okay, this is the W. W is a rough endoplasmic reticulum which is going to contain ribosomes attached which are involved in synthesis of proteins. So, here now X, okay, the X is the chloroplast. So, look into this, students, just it is a diagrammatic view. So, the X is a chloroplast, yes, and next one Z is a Golgi body, and next one Y is a mitochondria. Okay. Yes, these are just the rough diagrammatic views. I think you already have seen these particular cell organelles in the introduction class itself. So, you all have the basic idea how actually the mitochondria and chloroplast looks like. So, based on these all the labeling parts, here is the question. W is engaged in synthesis of proteins. Yes, this is the right statement. So, but here you need to understand the correct one. Okay, with regarding to this particular cell organelle, you need to understand the right option. So, here W is engaged in synthesis of proteins. The nucleus is continuous with the rough endoplasmic reticulum. So, when we are going to call it as rough endoplasmic reticulum, when it is going to contain the ribosomes attached to it. Okay. So, that is going to be involved in the synthesis of proteins. So, this is absolutely the right option. So, coming towards the second option, Z is present in both prokaryotes and eukaryotes. Please try to understand this particular statement. Yes, Z is present in both prokaryotes and eukaryotes. See what it is actually, Z is Golgi bodies. Okay. So, this is actually the irrelevant option. In latter it is 70S type, whereas in former it is of 80S type. Okay. So, in latter, in the sense in this, yes. So, in the eukaryotes it is 80S type of ribosomes, in the prokaryotes it is 70S type of ribosomes. Okay. First the primitive ones are the prokaryotes which is going to contain the 70S type, but here in the latter, in the sense in the eukaryotes he is saying that it is 70S type. So, it is the wrong one, and in the prokaryotes it is, in the eukaryotes it is the 80S type of ribosomes, but actually the Z here, okay, actually the Z is Golgi bodies. These are actually involved in just synthesizing and modification of proteins. So, it is just not involved, it is not related to the ribosomes only, students. So, but here the statements are wrong. Just I am explaining you, the eukaryotes are going to contain the 80S type of ribosomes, whereas prokaryotes are going to contain the 70S type of ribosomes. Next, coming towards the option 3, both X and Y are double membrane bound organelles. X is present only in the plant cells, whereas Y is present only in the animal cells. So, this is also the wrong. What is Y here? Mitochondria. Mitochondria is present in both plants and animal cells, but this statement is right. Y is present in both plants and animals. This is right, okay. But whereas coming towards the X, it is a chloroplast which is present only in the plant cells only, okay. Yes. So, this option is also the wrong, okay. Both are double membrane, it is right. X is present only in the plant cells, whereas Y is present only in the animal cells. Y is not only present in animal cells, it is also present in the plant cells also. So, this option is wrong. Coming towards the option 4, acrosome of the sperms developed by the modification and maturation of the eggs. So, this is also the wrong option, students, okay. So, this is related to the reproduction chapter. Here as if now according to this particular diagrammatic view and question, this will be the right option. W is engaged in synthesis of proteins. This will be the right option. Yes. Next, coming towards the next question. Yes. All the members of Gupta family can enroll their tongue, okay. All the members of the Gupta family can roll their tongue. Which of the parts labeled in the given figure carries the information regarding these characteristic, okay. So, it is just a characteristic, it is just one of the interesting trait of their family members, of their Gupta family members. Here are the labeling parts, okay. But in these labeling parts, there is only one part which is going to show this particular characteristic feature. So, you need to understand only that, okay. First, what is R here? It is the smooth endoplasmic reticulum. First, look into the labeling parts, okay. First, try to label the parts properly. And next, coming towards the option P, labeling part P, what it is? Nucleolus. So, this is just the stained spherical body inside the nucleus. And what is Q? The colored network like thread like structure present inside the nucleus is called as, already we have seen in the just now. See, these all colored network like thread like structures present inside the nucleus is called as, most important one, chromatin, chromatin material. So, during the cell division, actually the chromatins are going to, during the cell division process, they are going to be condensed and they are going to become thicker, okay, due to the dehydration process, and these chromatin material is going to contain the DNA, okay. They are going to contain the genetic information in the form of a segments called as DNA, and that DNA is extremely involved and it is extremely useful for the development and growth of the complete characteristics to their offsprings, okay. The genetic information mainly passes through this DNA only, okay, which is present inside the chromatin, okay. That DNA is involved, it is also involved in making and regulation of the proteins which are actually required for the cell and organism to grow. Yes. So, see here, what is P? P is nucleolus, okay. Nucleolus is a spherical body which is involved in making the ribosomes, but we cannot, from the nucleolus no genetic information can pass. And whereas coming towards the Q, Q is chromatin material. The chromatin contains the genetic information in the units called as DNA, and that chromatin is involved, okay. It is actually important for the complete development and growth of the particular individual, and from the by the help of this DNA and chromatin only the characters are going to, the traits are going to be inherited from the parents to their offsprings. So, this will be the right option. Coming towards the R and S, okay, these both are irrelevant options only. The S, this is all about the nucleoplasm. So, this is a wrong. And next one, R is smooth endoplasmic reticulum. This is going to be involved in the synthesis of lipids. Yes. So, here the right option is Q. Yes. Next question, identify the process shown in the given figure. So, here is just a hint given to you, semi-permeable membrane is must in the process. So, this is going to define the differentiation in between the osmosis and diffusion. Okay, you need to understand the concept of osmosis and diffusion, then it becomes easy to answer, students. See, option 1 is, it is a process that can occur in all the mediums. So, this is the wrong, it can occur, okay, it can occur only inside the solvent molecules only, okay, only solvent molecules are involved. Okay, it cannot occur in gaseous and liquid, gaseous and solid medium. Yes, so this option is wrong. So which option is right for these students? Which is going to occur in all the mediums? Diffusion. Okay, diffusion takes place in solids, liquids and gaseous medium, and all the molecules in different types of molecules are involved in diffusion. Oh, but only solvent molecules are involved in the osmosis. Yes, it is a process that involves flow of solute particles only. This is also the wrong statement. Okay, there is a no process related to the transportation of only solute molecules. Okay, solutes and solvents we have studied, but no process is involved in the transportation of only solutes. So this is wrong. So now let's look into the option 4. It helps in the exchange of gases, transpiration and photosynthesis. Yes, this is a right option, but it is related to the diffusion. But here you need to apply the hint called as a semi-permeable membrane to solve this question. So this is the right option, but here semi-permeable membrane is absent. But the right option is option 3. It is a process that is influenced by the number of solute particles dissolved inside the solvent. So this osmosis process is going to be, okay, solutes is going to be influenced by the solvent molecules. Okay, we are going to study both solutes and solvent transport mechanisms in the osmosis. So the option 3 will be the right option. Next one, yes, next question is refer to the given figure and select the incorrect statement regarding its labeled parts. So first you need to focus on the labeling parts. So what is that? It is the stroma. And what is X here? These are thylakoids, which are stacked upon one another, called as the grana. Yes, and coming towards the Y, what it is? Cell membrane. Yes, so clear with the labeling parts. So now here are the options. Y has carrier proteins that control the passage of molecules. Yes, so but you need to find the incorrect statement. So this option will be the right one, first one. Okay, Y is a cell membrane, it is involved in transportation of the molecules only. Okay, this will be the right one, right statement. Next, coming towards the option 2, Z is colorless granular substance containing a circular DNA molecule. What is Z here, students? It is a colorless granular structure. Z is a stroma which contains circular DNA molecule. Yes, so this is the right option. Coming towards the option 3, Z is a site of light reaction during the photosynthesis. We have seen just now that stroma is a liquid. Okay, it is a liquid which is going to be present inside the chloroplast. Stroma is involved in the dark reactions but not the light reactions. So the right option will be the option 3. Okay, so now let's have a look on option 4. X contains chlorophyll in its membrane. Yes, thylakoids are going to be involved in trapping the solar energy and they are involved in the process of photosynthesis. So all the options are right, but the option related to the stroma is wrong because it is involved only in the dark reactions, students, but not the light reactions. Yes. Coming towards the last question, refer to the given figures and select the correct statement regarding them. Yes. So first let's have a look on all these cell organelles. So it is a mitochondria. Yes. So it is a mitochondria, Q is vacuole, R is chloroplast, S is ribosome. So regarding this you need to solve the right option. T is double membrane bound cell organelle. Yes, that is true, mitochondria is double membrane bound. Whereas QR and S are single membrane bound. So this is the wrong option, students. Q is single membrane bound, but R is double membrane bound and S is non-membrane bound. Okay. So here you have to study the single membrane bond, double membrane bond and non-membrane bond. So chloroplast and mitochondria are double membrane bond, vacuole is a single membrane bond and ribosomes are non-membrane bond. Coming towards the second option, so this will be the wrong option. So coming towards second option, P is involved in synthesis of ATP, it is true, whereas S is involved in synthesis of proteins, this is also true. Yes. So Q is involved in the storage of proteins, whereas R is involved in the synthesis of proteins. Yes, Q is involved in storage of proteins. What is Q? So this is wrong. Okay, this is a vacuole which is going to contain the cell sap, whereas R is involved in synthesis of protein. This is true, but this is wrong. Okay, you always need to match the right pair. So this option will be the wrong option. Next, coming towards the option 4, R synthesizes lipids whereas S is involved in storage of lipids. What is R? It is a chloroplast. Okay, it is not involved in the synthesis of lipids, and S is involved in storage of lipids. What is S here? S is ribosomes. Okay, these are only involved in smooth endoplasmic reticulum. In the smooth endoplasmic reticulum we cannot find the ribosomes attached. Okay, so that is involved in synthesis of lipids. Okay, but here R synthesizes lipids. Okay, inside the chloroplast we can find the lipids also. Okay, but here this R is right, but this is wrong. So we need to match the right pair, that is first option. P is double membrane bond. Yes. Second option, students, P is involved in the synthesis of ATP, whereas S is involved in synthesis of proteins. Okay, this is the right option. So clear with all the concepts, clear how you this is the way how you need to attempt the diagram based questions, students. Okay, so mostly here you need to focus upon the labeling parts, its functions, and you need to learn about each and every cell organelle completely, and you also need to practice the diagram, students. Okay, this is all about the foundation series class 2, where we have covered all the most important diagram based questions. And in the next upcoming foundation series we are going to look into some important and interesting things of some mind maps and some other concept oriented questions, students. So please stay tuned and like, share and subscribe to our channel NEET World. Thank you.